What if one pair of organs controlled not just fertility, but the entire course of male development from life in the womb to adulthood? Today, we uncover the hidden power of the testes, the body's dual factory and command center. Why does understanding these organs matter? Because whether you're treating a teen with delayed puberty, a man struggling with infertility, or a patient facing hormone imbalances, testicular health is at the core of male medicine. In this session, we'll break down development, the genetic signals that determine a person's biological destiny. Function, how sperm and testosterone are made. Pathology, what goes wrong, how to spot it, and how to treat it. Ready to master the knowledge that can change your patients' lives and ace your ASCP exam? Let's get started. Our story of the testes begins long before they even exist, in the very early stages of embryonic life. It starts with a special group of cells known as primordial germ cells. Think of these as the fundamental ancestors of all future sperm. Initially, these cells are not located where the testes will eventually form. Instead, they appear in a region outside the embryo itself, in a structure called the yolk sac. From this distant location, they must embark on a remarkable journey. Around the third week of development, these cells begin to migrate, moving with purpose through the developing embryonic tissues. Their destination is a pair of structures called the gonadal ridges, which are the primitive precursors to the gonads. This migration is a critical and precisely timed event in development. The primordial germ cells travel along the gut of the embryo, eventually arriving at the gonadal ridges by the 6th week. Imagine this as a carefully choreographed cellular march essential for the future of reproduction. Once they arrive, these germ cells don't remain isolated. They become enveloped by other cells originating from the ridges known as primitive sex cords. This combination of migrating germ cells and local ridge cells forms the very first basic structure of the primitive gonads. This process is identical in all embryos at this stage, whether they are genetically destined to be male or female. It's a foundational step that sets the stage for everything to come. The successful completion of this journey is non-negotiable for normal reproductive development. If the primordial germ cells fail to reach the gonadal ridges, the gonad simply will not develop properly. They would become what is known as gonad, a structure lacking the germ cells necessary to produce sperm or eggs. This highlights just how crucial these early cellular movements are. The entire potential for future fertility rests on this microscopic migration. The formation of these primitive gonads marks the end of the first phase, establishing the raw material from which either testes or ovaries will be sculpted. The stage is now set for a pivotal decision at this point. Around the 6th week of gestation, we have a pair of indifferent or bipotential gonads. They hold the potential to develop along either the male or the female pathway. The structure is ambiguous, and the embryo's ultimate direction has not yet been determined by the gonads themselves. The primordial germ cells are nestled within the sex cords, waiting for instructions. The next act in this developmental drama involves a genetic trigger that will push this neutral structure down one of two distinct paths. This is the moment where the genetic blueprint of the embryo begins to assert its influence, initiating a cascade of events that will define the individual's biological sex. We have now reached a critical juncture in development. The primitive gonads are formed, but they are in a state of neutrality. This is the bipotential stage. The term bipotential simply means having two potential pathways of development. At this point, the gonad is anatomically identical in both 46XX genetically female and 46XY genetically male embryos. It contains all the necessary components to become either a testis or an ovary. Think of it as a piece of uncarved stone that a sculptor can shape into one of two different statues. The internal architecture of the gonad has two main parts: an outer region called the cortex, and an inner region called the medulla. This dual structure is key to its flexibility. In the absence of specific male directing signals, the gonad will automatically follow the female developmental pathway. The cortex will develop and mature, eventually forming an ovary, while the medulla will regress and fade away. This is considered the default path in human development. For the male pathway to be taken, an active intervention is required. Something must step in and override the default female program. This is a fundamental concept in sexual differentiation. The formation of testes requires a specific genetic push. Without this push, the bipotential gonad will passively develop into an ovary, regardless of the embryo's chromosomal makeup. This makes the male developmental signal incredibly important. This fork in the road is governed by genetics. The decision of which path to take hinges on the presence or absence of a single chromosome, the Y chromosome. If the embryo is 46XX, it lacks the Y chromosome and therefore lacks the critical male directing signal. As a result, the default ovarian pathway proceeds. The cortical sex cords will develop surrounding the germ cells to form ovarian follicles. Conversely, if the embryo is 46XY, it possesses the Y chromosome. This chromosome carries the master switch gene that will reroute development away from the ovarian path and actively steer it toward forming a testis. This genetic instruction is the key that unlocks the male developmental program. So, the bipotential gonad sits and waits for its marching orders. Its fate hangs in the balance, entirely dependent on a genetic signal. The embryo has two sets of primitive ducts developing alongside the gonads, the Müllerian ducts, which are female precursors, and the Wolffian ducts, which are. The fate of these ducts is also tied to the gonad's development. The decision made by the gonad will determine which set of ducts thrives and which one withers away. The entire reproductive anatomy, both internal and external, is waiting for this first, crucial choice. The bipotential stage is a fleeting but profoundly important moment where genetics dictates destiny. The master switch that diverts the bipotential gonad toward the male pathway is a gene located on the short arm of the Y chromosome. This gene is called the SRY gene, which stands for sex-determining region Y. For many years, it was referred to by the placeholder name TDF, or testis-determining factor, before the specific gene was identified. The SRY gene produces a protein that acts as a transcription factor. This means its job is to bind to specific regions of DNA and control the activity of other genes. The SRY protein initiates a cascade of genetic events, a sort of domino effect that commands the cells of the bipotential gonad to organize into a testis. When the SRY gene is activated in a 46XY embryo, its protein product goes to work inside the primitive gonad. It triggers the cells of the medullary cords to differentiate and proliferate. These cords develop into the seminiferous tubules, the future sperm-producing factories of the testes. Another crucial cell type, the Leydig cells, also begins to form in the tissue between these developing tubules. These Leydig cells are critically important because they will soon begin producing androgens, the male hormones. At the same time, another specialized cell type, the Sertoli cells, develops within the walls of the seminiferous tubules, providing support for the germ cells. SRY is the spark that ignites this whole process. The activation of the SRY gene effectively suppresses the female developmental program. Instead of the cortex developing into an ovary, it regresses. The medulla, which would have regressed in a female embryo, now takes center stage and forms the functional tissue of the testis. This is a clear and active diversion from the default pathway. The primordial germ cells, which have been waiting patiently, are now incorporated into the newly forming seminiferous tubules. Here, they will become known as spermatogonia, the stem cells that will eventually undergo mitosis and meiosis to produce sperm. Though this process will not begin until puberty, many years later. By about the 7th week of gestation, the effects of the SRY gene are morphologically visible. The gonad is no longer indifferent, it has differentiated into a recognizable fetal testis. Almost immediately, this newly formed organ begins its endocrine function. Stimulated by human chorionic gonadotropin, or HCG, a hormone produced by the placenta, the fetal Leydig cells start to secrete testosterone. This is the first major hormonal step in male development. The production of testosterone by the fetal testes will in turn direct the development of the male internal ducts and external genitalia, completing the next phase of sexual differentiation. The genetic switch has been flipped and the male pathway is now fully engaged. Once the SRY gene has done its job and a fetal testis has formed, the next critical phase of male development begins. This phase is driven not by genes directly, but by the hormones produced by the newly formed testes. Remember those two sets of primitive ducts present in every early embryo. We have the mesonephric ducts, also known as Wolffian ducts, which are the precursors to the male internal reproductive tract. And we have the paramesonephric ducts or Müllerian ducts, which are the precursors to the female internal tract in a male embryo. The fetal testes must now send out powerful hormonal signals to ensure the correct set of ducts develops, while the other one disappears completely. The first key hormone in this process is testosterone, which is produced by the Leydig cells of the fetal testes. Testosterone acts directly on the Wolffian ducts. It stimulates them to survive, grow, and differentiate into the essential components of the male internal plumbing. Under the influence of testosterone, the Wolffian ducts develop into the epididymis, the vas deferens, and the seminal vesicles. These structures are responsible for transporting, maturing, and storing sperm later in life. Without this testosterone signal, the Wolffian ducts would simply wither away and vanish, as they do in a female embryo. This hormonal support is an absolute requirement for building the male internal reproductive system. However, simply developing the male ducts is not enough. The female precursor ducts, the Müllerian ducts, must be eliminated. This task falls to a different set of cells within the fetal testes, the Sertoli cells. These cells produce a crucial substance called anti-Müllerian hormone, or AMH. As its name suggests, AMH actively causes the Müllerian ducts to break down and regress. This is an active process of destruction, preventing the formation of a uterus, fallopian tubes, and the upper part of the vagina. The coordinated action of testosterone and AMH is therefore essential. One hormone builds the male structures while the other demolishes the potential female structures. This two-hormone system ensures that the internal reproductive anatomy develops unambiguously along the male line. In a female embryo, which lacks testes, there is no testosterone to support the Wolffian ducts, so they regress. There is also no AMH to destroy the Müllerian ducts, so they are free to develop into the female internal tract. This highlights a critical principle. The development of the male internal tract is an active hormone-dependent process. It requires both the presence of testosterone and the presence of AMH. Any disruption in the production or action of these hormones can lead to significant anomalies in sexual development. With the internal reproductive tract set on the male path, the final step in embryonic sexual differentiation is the development of the external genitalia. In the early embryo, just like the internal ducts, the external structures are undifferentiated and identical in both sexes. These structures consist of a small bump called the genital tubercle, a pair of urethral folds, and a pair of labioscrotal swellings. From this common starting point, these structures can develop into either male or female external genitalia. The deciding factor once again is the presence or absence of powerful male hormones, specifically androgens. This process occurs slightly later than the differentiation of the internal ducts. The primary hormone responsible for this masculinization is not testosterone itself but a more potent derivative called 5-alpha-dihydrotestosterone, or DHT. In the target tissues of the external genitalia, an enzyme called 5-alpha reductase converts testosterone into DHT. This supercharged androgen is what drives the dramatic changes that form the male anatomy. Under the influence of DHT, the genital tubercle elongates and grows significantly to form the glands of the penis. The urethral folds fuse along the midline to enclose the urethra, forming the shaft of the penis. Finally, the labioscrotal swellings expand and fuse together to create the scrotum, which will eventually house the testes. This conversion of testosterone to DHT is a crucial step. The tissues of the external genitalia are uniquely sensitive to DHT, and testosterone alone is not potent enough to complete this transformation fully. This is why a deficiency in the 5-alpha reductase enzyme can lead to a condition where an individual is genetically male, 46XY with functional testes and normal internal male ducts, but is born with ambiguous or female-appearing external genitalia. It powerfully illustrates the specific and essential role that DHT plays in sculpting the final external form. The entire process is a testament to the power of hormones to direct development down a specific path. In the absence of androgens, as in a female fetus, these same undifferentiated structures follow the default female pathway. The genital tubercle develops into the clitoris, the urethral folds remain separate and become the labia minora, and the labioscrotal swellings develop into the labia majora. This outcome occurs regardless of whether an ovary is present. It is simply the path taken when high levels of androgens are not there to push development in the male direction. By the end of the first trimester, the three major steps of male sexual differentiation are complete. The gonads have become testes, the internal ducts have formed, and the external genitalia have been masculinized. After birth and following the transformative period of puberty, the testes are fully mature and functional. The adult testes are a pair of ovoid organs, each typically measuring about 4 to 5 centimeters in length. They are housed within the scrotum, which is a muscular sac of skin located outside the main body cavity. This external location is not accidental. It's actually a critical design feature. The production of sperm or spermatogenesis is highly sensitive to temperature. It requires an environment that is approximately 2 degrees Celsius, cooler than the core body temperature. The scrotum acts as a climate control system, using muscles like the dartus and cremaster muscles to raise or lower the testis, moving them closer to or further from the body to maintain this optimal temperature. Each testis is protected by a tough, fibrous outer capsule called the tunica albuginea. Internally, the testis is divided into about 250 to 300 small compartments known as lobules. Within each of these lobules lies the functional machinery of the organ. The bulk of the testis is comprised of a vast network of tiny coiled tubes called the seminiferous tubules. If you were to unwind and lay out all the seminiferous tubules from a single testis, they would stretch for hundreds of meters. It is within the walls of these tubules that spermatogenesis takes place. The tubules contain the developing germ cells at various stages of maturation, alongside the supportive Sertoli cells that nourish and guide them. The tissue that fills the spaces between the seminiferous tubules is called the interstitium. This interstitial tissue is also critically important because it is home to the Leydig cells. These are the cells responsible for the second major function of the testes, producing testosterone. They are strategically located near a rich network of blood vessels, which allows the testosterone they produce to be efficiently secreted directly into the bloodstream and distributed throughout the body. Therefore, the testis has two distinct anatomical and functional compartments, the tubules for sperm production and the interstitium for hormone production. These two compartments work in concert, communicating via local hormones to regulate their respective functions. Once sperm are produced in the seminiferous tubules, they are not yet mature or capable of swimming. They are transported out of the testis through a series of ducts, beginning with the reedy testis and then the efferent ductules. These ducts lead into the epididymis, which is a single, long, tightly coiled tube that sits on the posterior side of each testis. The epididymis serves as a finishing school and storage site for sperm. Here they spend several weeks undergoing final maturation, acquiring motility and the ability to fertilize an egg. From the epididymis, mature sperm move into the vas deferens, ready for their journey out of the body during ejaculation. The first primary job of the adult testes is to function as a highly efficient and continuous factory for producing sperm. This remarkable biological process is called spermatogenesis. It begins at puberty and continues uninterrupted throughout a man's adult life well into old age. The entire process takes place within the walls of the seminiferous tubules. The starting materials for this factory are the spermatogonia, which are the resident stem cells of the testes. These cells are the direct descendants of the primordial germ cells that migrated into the gonads during embryonic development. They lie dormant throughout childhood, waiting for the hormonal signals of puberty to awaken them and begin their lifelong task of sperm production. Spermatogenesis is an incredibly orderly and lengthy process, taking roughly 74 days from start to finish. It begins when a spermatogonium divides. One of the resulting cells remains a stem cell, ensuring the factory never runs out of raw materials. The other cell is committed to becoming sperm and embarks on a journey of transformation. This cell undergoes a series of divisions, including both mitosis and, crucially, meiosis. Meiosis is the special type of cell division that reduces the number of chromosomes by half. This is essential because the final sperm must be haploid, containing only 23 chromosomes, so that it can combine with a haploid egg to form a new, complete 46-chromosome individual. Throughout this entire maturation process, the developing sperm cells are not alone. They are physically supported and nourished by the large Sertoli cells that form the walls of the seminiferous tubules. The Sertoli cells are often called nurse cells because they perform many vital functions. They create a protected environment for the developing sperm, shielding them from the bloodstream. They provide nutrients, remove waste products, and secrete fluids that help transport the sperm. They also produce important signaling molecules that regulate the progression of spermatogenesis. The developing germ cells are embedded within these Sertoli cells, moving slowly toward the central lumen of the tubule as they mature. Finally, after completing meiosis, the haploid cells, now called spermatids, must undergo a dramatic physical transformation in a process called spermiogenesis. They are still simple, round cells and do not yet look like the familiar swimming sperm. During this final stage, they shed most of their cytoplasm, grow a long tail or flagellum for motility, and develop a specialized head. The head contains the compacted genetic material and is capped by the acrosome, a small packet of enzymes needed to penetrate the outer layers of an egg. Once this transformation is complete, the mature-looking but still immotile sperm, now called spermatozoa, are released from the Sertoli cells into the lumen of the tubule, ready to begin the next stage of their journey. The second critical function of the testes is to act as the body's primary hormone production hub for androgens. This process is called hormonogenesis, and the star player is testosterone. While the adrenal glands produce a small amount of androgens, the vast majority of testosterone in a male is synthesized by the Leydig cells, which are located in the interstitial tissue between the seminiferous tubules. This hormonal production is just as important as sperm production, as testosterone is required not only for spermatogenesis itself, but also for maintaining the entire male physique and function, from muscle mass and bone density to libido and secondary sexual characteristics. The production of testosterone is tightly controlled by a sophisticated feedback system involving the brain. The process begins in the hypothalamus, which releases gonadotropin-releasing hormone, or GnRH, in a pulsatile fashion. This GnRH travels to the pituitary gland and stimulates it to release two key hormones, luteinizing hormone, or LH, and follicle-stimulating hormone, or FSH. Although named for their roles in the female menstrual cycle, these hormones are essential in males. LH is the primary signal that acts on the Leydig cells in the testes. It binds to receptors on the surface of these cells and triggers the biochemical cascade that leads to the synthesis of testosterone. The raw material for making testosterone is cholesterol. Leydig cells can either synthesize their own cholesterol or, more efficiently, pull it from the bloodstream. Once inside the cell, a series of enzymatic reactions, initiated by the LH signal, converts cholesterol step by step into testosterone. This newly synthesized testosterone is then released into the bloodstream, where it can travel to target tissues all over the body. It is also secreted locally within the testes at very high concentrations. This incredibly high local level of testosterone is absolutely essential for the Sertoli cells to support the process of spermatogenesis occurring in the adjacent tubules. Without it, sperm production would fail. To prevent testosterone levels from getting too high or too low, the body uses a negative feedback system. When testosterone levels in the blood rise, this signals back to both the hypothalamus and the pituitary gland, telling them to reduce the secretion of GnRH and LH. This in turn, reduces the stimulation of the Leydig cells, and testosterone production slows down. Conversely, if testosterone levels fall, the negative feedback is reduced, and the hypothalamus and pituitary increase their signals to ramp up production. This elegant loop ensures that testosterone levels are kept within a relatively stable range, maintaining hormonal balance throughout the body. Sperm begin their journey in the seminiferous tubules, but at this stage they're immobile and unable to fertilize an egg. They travel through a network of channels, tubuli recti, rhytate testis, and efferent ductals before entering the epididymis. The epididymis, a long coiled tube, is where sperm mature over several weeks, gaining motility and the ability to fertilize. Once mature, sperm are stored in the tail of the epididymis and the start of the vas deferens, remaining viable for weeks. During ejaculation, muscular contractions propel sperm through the vas deferens into the ejaculatory ducts. Here, sperm mix with fluids from the seminal vesicles and prostate gland, forming semen. Seminal vesicle fluid provides energy, while the prostate's alkaline secretion protects sperm in the acidic vaginal environment. The final mixture, semen, is expelled through the urethra during ejaculation. Testicular function is governed by the hypothalamic-pituitary-testicular axis, a precise hormonal command chain. The hypothalamus releases GnRH impulses, signaling the pituitary to secrete LH and FSH. LH stimulates Leydig cells to produce testosterone. FSH acts on Sertoli cells to support sperm development. Testosterone and FSH together ensure proper sperm maturation. Rising testosterone levels send feedback to the hypothalamus and pituitary, reducing GnRH and LH release, a classic negative feedback loop. Sertoli cells release inhibin, which specifically inhibits FSH, fine-tuning sperm production. This two-way communication keeps the system balanced and responsive. Disruption at any level can impact fertility and hormone levels. The HPT axis integrates brain and testicular function for reproductive health. Testosterone's influence begins in the womb, shaping male reproductive organs and external genitalia. After birth, testosterone production pauses until puberty, when the HPT axis reactivates. Puberty brings a surge in testosterone, driving growth of the testes, penis, muscle mass, and secondary sexual traits. In adulthood, testosterone maintains libido, erectile function, sperm production, bone density, and energy. Levels fluctuate daily, peaking in the morning and declining by evening. This rhythm is important for clinical testing and reflects the dynamic nature of hormone regulation. Testosterone's effects extend beyond reproduction, supporting overall male health. Its lifelong presence is essential for both development and maintenance. Puberty marks the awakening of the male reproductive system, starting with testicular enlargement due to rising FSH and LH. Testosterone drives physical changes, genital growth, hair development, voice deepening, and the pubertal growth spurt. Growth plates eventually fuse, setting adult height. Delayed closure can occur with hormonal disorders. Aging brings a gradual decline in testosterone, sometimes called andropause. Increased SHBG with age further reduces active testosterone. Symptoms of low testosterone in older men include muscle loss, reduced bone density, decreased libido, and mood changes. Loss of secondary sexual traits and late-onset hypogonadism may occur. Testosterone's decline impacts health and quality of life in aging men. Genetic and congenital disorders can disrupt testicular development and function from birth. Klinefelter's syndrome 47XXY causes small testes, low testosterone infertility, and sometimes learning difficulties. Androgen insensitivity syndromes occur when cells can't respond to testosterone, leading to female-appearing external genitalia in genetic males. Testicular feminization syndrome is the most severe form, with individuals raised as girls but lacking a uterus or ovaries. 5-alpha reductase deficiency impairs DHT production, resulting in ambiguous genitalia at birth and virilization at puberty. Some systemic genetic disorders, like myotonic dystrophy, cause testicular failure later in life. This leads to infertility, low testosterone, and elevated FSH and LH. Testicular health is closely linked to overall genetic and systemic health. Early diagnosis and management are crucial for affected individuals. These conditions highlight the complexity and vulnerability of male reproductive development. Acquired testicular dysfunction, causes and consequences. Beyond congenital and genetic conditions, the tests can also be affected by problems that are acquired later in life. These issues can stem from a wide variety of causes including direct injury, infections, systemic diseases, and of course, even the natural process of aging. One well-known cause of testicular damage is infection. The mumps virus, if contracted after puberty, can lead to a painful inflammation of the testes called mumps or chetes. In a significant number of cases, this can result in permanent damage to the seminiferous tubules, leading to testicular atrophy and impaired fertility. Other infections, including certain viruses and bacteria, can also cause orchitis with similar consequences. Moreover, treatments for other diseases, such as chemotherapy and radiation for cancer, can be highly toxic to the rapidly dividing germ cells in the testes, often causing long-term or permanent infertility and hormonal deficiency. Systemic diseases can also have a profound impact on testicular function, often by disrupting the hypothalamic-pituitary-testicular axis. This leads to a condition known as hypogonaditropic hypogonadism, where the problem isn't with the testes themselves, but with the signals they receive from the brain. A classic example is hyperprolactinemia, or high levels of the hormone prolactin, often caused by a small tumor on the pituitary gland. Elevated prolactin can suppress the pulsatile release of GnRH from the hypothalamus, leading to low LH and FSH, and consequently, low testosterone and impaired sperm production. Similarly, type 2 diabetes is strongly associated with hypogonaditropic hypogonadism in men, likely through a combination of factors including insulin resistance and chronic inflammation that disrupt the normal signaling from the pituitary. The natural process of aging, as we've discussed, is another major cause of acquired testicular dysfunction. The gradual decline in testosterone production that begins around age 30 can eventually lead to clinically significant hypogonadism in older men. This is often a combination of both primary testicular decline and secondary dysfunction from a less robust hypothalamic-pituitary response. When an older man presents with symptoms like fatigue, low libido, loss of muscle mass, and bone density loss, it is crucial to investigate his hormonal status. The diagnosis of hypogonadism requires both the presence of these clinical signs and symptoms and corroborating biochemical evidence, namely, consistently low morning testosterone levels measured on more than one occasion. Identifying the cause of testicular dysfunction is a critical diagnostic step. The first thing a clinician will do is distinguish between primary hypogonadism, which is testicular failure, and secondary hypogonadism, which is pituitary or hypothalamic failure. This is easily done by measuring serum levels of LH and FSH. In primary hypogonadism, the testes are failing, so the pituitary gland works overtime to stimulate them, resulting in high levels of LH and FSH. In secondary hypogonadism, the problem is in the brain, so LH and FSH levels will be inappropriately low or normal despite low testosterone. This simple blood test points the investigation in the right direction, toward the testes in the first case, or toward the pituitary and hypothalamus, possibly requiring brain imaging like an MRI, in the second. Bringing it all together, testicular health, treatment, and lifelong care. Our comprehensive journey has revealed the testes as remarkable organs, central to male health from embryonic development through to old age. We've seen how a precise genetic switch initiates their formation, how a symphony of hormones orchestrates their function, and how they perform the dual roles of sperm and testosterone production. Understanding this intricate biology is fundamental because testicular health is a powerful indicator of overall well-being. The signs and symptoms of testicular dysfunction, from delayed puberty and infertility to fatigue and bone loss, are important clinical clues that demand attention. Recognizing these issues early and initiating a proper diagnostic workup is a critical skill for any physician, as it opens the door to effective management and can significantly improve a patient's quality of life. When the diagnosis of hypogonadism is confirmed with both consistent clinical symptoms and verified low testosterone levels, testosterone replacement therapy becomes a primary treatment option. The guiding principle of this therapy is straightforward: to restore serum testosterone concentrations to the normal physiological range, thereby alleviating the symptoms of deficiency. Several methods of administration are available, each with its own profile of benefits and drawbacks. The most common and cost-effective method involves ultramuscular injections of testosterone esters, such as cypionate or enanthate, typically given every 1 to 2 weeks. This method is effective but can lead to fluctuations in hormone levels, with peaks after the injection and troughs before the next one is due, which some patients may find noticeable. To provide more stable physiologic hormone levels, transdermal preparations were developed. These include patches worn on the skin, or more commonly today, hydroalcoholic gels that are applied daily. These methods deliver a steady amount of testosterone throughout the day, mimicking the body's natural rhythm more closely. However, they require daily application and carry a risk of unintentional transfer to others, such as a female partner or children, through skin-to-skin contact, which requires careful precautions. Other options, like buccal systems or long-acting subcutaneous pellets, offer alternative delivery mechanisms, but may have their own limitations regarding convenience, cost, or local side effects. The choice of therapy should be a shared decision between the clinician and the patient, considering lifestyle, cost, and personal preference. Once testosterone therapy is initiated, diligent monitoring is absolutely essential to ensure both efficacy and safety. The goal is to maintain testosterone levels in the mid-normal range and to assess for clinical improvement. However, it is equally important to screen for potential adverse effects. Before starting and periodically during treatment, physicians must monitor the patient's hematocrit, as testosterone can stimulate red blood cell production and potentially lead to polycythemia and abnormally high red blood cell count. Prostate health must also be watched closely. While testosterone therapy does not cause prostate cancer, it can stimulate the growth of a pre-existing, undiagnosed cancer. Therefore, monitoring prostate-specific antigen or PSA levels and performing digital rectal exams are standard practice, especially in older men. This vigilant follow-up ensures that the benefits of therapy continue to outweigh the risks throughout the patient's life.